Welcome to Inside Bioprocessing, a Thermo Fisher scientific podcast discussing key trends and topics within the bioprocessing industry. In this episode, we will be discussing optimizing stable clone development to improve biopharmaceutical production. Biologics are a growing part of the healthcare industry, and as such, there is a continued drive from developers to produce high-quality products that are safe, effective, and economically feasible to manufacture. Cell line development is an early and crucial part of this development journey and is important to get right. However, the process of developing a stable clone can be challenging and time-consuming. To discuss the current state of stable clone development and explore how developers can successfully integrate them into their commercial manufacturing process, we are joined today by two experts working in the field. Please join me in welcoming Garima Mishra, Field Application Staff Scientist for Cell Culture and Cell Therapy at Thermo Fisher, and Volker Sandig, Chief Scientific Officer at ProBioGen. Garima, could you begin by explaining the importance of stable clone development and outline the key steps of the development process, from initial plasmid development through to the creation of the final stable clone used for manufacturing?
Biologics are becoming an essential branch of the healthcare industry. Biopharmaceutical manufacturing employs living host cells as protein production workhorses. Cell line development is the starting point of a biologics developmental journey. Development of these cell lines is a complex, multi stage, time consuming process requiring specific expertise and access to suitable technologies. A typical cell line development workflow consists of expression vector creation, carrying gene of interest, cloning and stable transfections, single cell cloning, clone screening, and clone selection, followed by cell line characterization, expansion, and cell banking. Historically, the main center of attention in cell line development has been on generating and finding highly expressing clones through optimizing selection systems, expression vectors, and screening. Lately, on the concept of process defines the product, development of chemically defined media and feeds has resulted in the better understanding of nutrient influence on product quantity and quality. Current process productivities are a combination of an effective protein expression system, a good host cell line, selection of a suitable product variant, appropriate media feeds, and process operating conditions.
So, Volker, with achieving optimal results in mind, what would you say are the defining attributes of a CGMP-ready stable clone?
When you develop a CGMP-ready cell line, you need to monitor your process much better. You have to record the full history of your cell line because that has to be provided to regulators. This also applies to a cloning history. You need to show that your cell line is indeed clonal, and you have to provide proof for it. This may be challenging sometimes. Finally, you need to make sure that you don't achieve a good yield once. You have to provide a robust, process-ready cell line that achieves final cell densities and good viabilities in the end of a process each time you run a process at multiple scales. Tide, of course, plays a role as this defines an economic production process, but you also need to confirm product features other than titer. This applies to post-translational modifications like the sugars, glycans, but also deamidation, oxidation, and so on, which may be sometimes clonal features. One of those is aggregation that really depends on the clone. You have to make sure this clone is stable, and this applies not only to the yield in the process, but also to copy numbers of integrated plasmids and the amount of RNA synthesized in the production phase of a fat batch for each of the chains of your molecule. Finally, there is freezing stability. You need to make sure these features don't change when you lay down a master cell bank and start a production process thereafter. This altogether defines your GMP-ready cell line.
That's really interesting. Thank you. So, in terms of attaining those attributes, what are the typical challenges that researchers and manufacturers come up against when developing a stable clone?
These challenges actually start from selecting a vector and selecting a host cell line. This host cell line needs to have a long history. It needs to have a serum-free history. Otherwise, you cannot prove that there is no issue with adventitious agents and prion diseases. It continues with efficient transfection. Not every host will support that, and yet transfection efficiency is critical for final yields. There is when you produce a GMP-ready cell line, you had one before, or you had product before. It was either generated in transient transfection, or in a clone pool, or in an earlier cell line. Now you need to match product features between the two, and that is a challenging thing if you change systems, because often the effort in cell line development is doubled when you have differences and have to tweak product features to the originally observed ones in order to match product activity, and you have to do that by process development. This costs you lots of time. It is also critical that your starter cell line does not change anymore, because a changing cell line would result in instability later on. From the vector side, it is critical that your vectors support multiple formats of potential products, ratios between the respective chains, and a selection that finally allows you to provide stability, because it has a moderate number of integrated copies and is not amplified, because amplification often leads to instability. Finally, the medium plays a crucial role in this process. The medium defines the properties of the cell line in terms of stability. It also determines post-translational modifications. So keeping that from an earlier process is highly recommended.
One thing that you mentioned there that would be really interesting to hear a little bit more about is vector systems. Could you outline any best practices for designing and optimizing a vector system for recombinant protein production?
There's quite a variety of vector systems used in the industry to make cell lines. The two basic concepts are that you either put all all genes of the respective product on a single vector. In that case, you need a single selection marker. And this is a robust system that is used widely. It's simple. However, besides increasing the time a little bit for the cloning effort, the DNA cloning effort, it is rather restricted in terms of ratios of expression levels between chains. Even as, like for a simple antibody, you try to give more benefit to a light chain because the heavy chain is only secreted in the presence of the light chain. However, this may vary among antibodies, so you would ultimately like to change chain ratios. This is even more the case for a complex molecule that consists of more than two chains and where a perfect pairing, an equimolar pairing, is required. In this case, a single vectors for each chain are preferred, and so you have a set of vectors to be transfected, and this requires multiple selection markers. These vectors need to be equipped with strong promoters. They also need protective elements that save the genes from epigenetic silencing, DNA methylation, and histone methylation deacetylation. All those elements need to be on the vector and together with selection markers. And if these selection markers are highly expressed, They take resources of the cell just for a selection marker, which is there to create the cell line but not to produce. So often care is taken to connect those selection markers with weak promoters. This is something we did here for the kit. There is, of course, more you can do to vectors. You can use like a locus control regions, larger regions, in order to provide expression stability. But like each system here has its benefits, and it is critical to have a well-adjusted one, as we do for this kit.
Just to look a bit further along the process development workflow towards research and commercial production, Garima, why is it important to fully understand and characterize a stable clone before moving on to these later stages?
So, the development of a robust, highly yielding, stable cell line is the foundation of any reliable production process. And we have heard from Volker that it is super crucial to have a well established host cell line, vector system, media, feed, and other processes. Cell line characterization is a critical part to the control of biological product manufacturing. The main objective of this testing to characterize a cell line is to confirm the identity, purity, suitability of the cell substrate for manufacturing these biologics. And as these cell lines express proteins constantly, there is a challenge to get the desired protein in large quantities as fast as possible and with the right quantity. It is a regulatory requirement that cells producing these therapeutic proteins are derived from a single cell clone. It is a well established thought process in the industry which allows the management of cell line heterogeneity arising from the well known cho-genome plasticity. Regulatory documents like ICH-Q5D provides guidance on standards for the derivation of cell lines to be used in biotechnological or biological products about the origin, source, and history of cells. All these steps are important to make the regulators convinced about the generation of these parental cell lines from the single progenitor cells. So it's super crucial to characterize a stable cell completely before moving on to the research or commercial production stage.
So, once a suitable clone has been established, then, how can this be successfully scaled up and integrated into a commercial manufacturing process?
So once a regulatory friendly CGMP stable clone is identified, established, characterized, the goal of a developer is to reach GMP manufacturing rapidly for either IND submission or for continuously supporting clinical and commercial manufacturing. It is important to adopt the automation and high throughput platforms from the beginning of these biotherapeutic developments, as this can streamline the scale up to commercial manufacturing by facilitating the assessment of multiple parameters in parallel, where conditions can be maintained from research scale to commercial scale. And there have been exciting scientific developments in CLD. One example is, for example, using a platform basal production media, which will enable a connect between clone and the process development, and also allow a developer to reduce the risk which might be associated with early stage process development, and thereby reducing the timelines. As I mentioned, that platform based basal media can be one of the approaches which allows the seamless scale up from the research to the commercial manufacturing, and these medias in conjugation with specific feeds will allow delivery of superior product performance, improved product titer, better product quality, and lot to lot consistency, which is one of the requirement for a controlled and have predictability at commercial scale.
Perhaps I wanted to make a small addition to the question. So, as you, Garima, outlined, the fit between a cell line and the process is important, and the process development is an essential part of cell line development, and it can take more time even than making the cell line itself. With the kit, we have taken the approach to do this work for you, for a new cell line development before you even start it. We have established a standard process and we try to find a clone that fits that process. This is really the time saving, and it is also the way to a really industrial process early on. We understand, of course, that this process has to be refined when you go to late stage phases. But these are small modifications, perhaps guided by a multitude of processes and digital analysis, artificial intelligence like digital twins that guide you to an even better process, but on the basis of what has been developed already.
That's a really interesting point. From what we've heard so far, it's clear that the stable clone development process can be complex. So, what expertise and solutions are available for researchers and manufacturers who may be experiencing development challenges?
So at Thermo Fisher Scientific, we understand the challenges of early phase development and recognize that each project may have unique challenges and targeted outcomes. We have GIPCO PD Express services, which has experience necessary to solve cell line media process development related challenges. The GIPCO Freedom expicho-s kit is one of the examples that provides the components and workflow solutions needed for a successful transfection, expression, clone selection, stable cell line generation, plus flexible royalty-free licensing options. Also, we have learned that every cell may have different nutritional needs, and media optimization will lead to improved titers and productivity, which in turn save time and money. GIPCO Media Solutions services are apt for every budget and timeline, leveraging expertise and experience. Our flexible media development options now include new and proprietary multi-omics workflow, which allows a client to characterize process to improve productivity. So there are panel of services available to troubleshoot and tackle these challenges, which might come along with the development of these stable clones.
You mentioned the GIBCO Freedom expicho-s kit there, which is the latest addition to the GIBCO Freedom Kit family. Volker, could you outline how this new kit differs from previous iterations?
Yeah, in 2011, we launched the Freedom cho-s kit, also co-developed between ProBioGen and Thermo Fisher. This kit was a great success, but of course it delivered titers which were normal and very good at the time. This kit now is a substantial improvement on several levels. First, we have taken again a proprietary, good, well-known cell line forward. But instead of the cho-s, the expicho cell line that is known for its superior features in transient transfections. Using the same cell line in transient and stable cell lines assures that the products are the same. We have the same host. Moreover, the media platform has been refined specifically for the kit, and this applies in particular to the feed. Of course, such a cell line has higher transfection efficiencies because it naturally does if it's suited for transient transfection. We have also done quite a bit on the vector. In the first kit, in the cho-s kit, a single vector was used for all chains. It performed great for some molecules, but not for all. Now we have separated the genes on separate vectors, which provides this flexibility we need for next generation biologics. We also have improved on promoters. We have again included introns and a stabilizing element. And we are using the same selection markers as we did before. But we take care that we have a general process applicable by everyone with simple tools also for cloning and protocols and a workflow that is manageable by all without a very specific sophisticated equipment. So it is a generally usable kit that now delivers like much higher titers of 3 to 5 grams per liter, which is just perfect and today's standard.
And finally, to round off the podcast, we wanted to get your thoughts on the future of stable clone development. So, in both of your opinions, what do you see as the most exciting developments in this space, both at research and industrial scale? Volker, would you like to go first?
There's actually many such exciting developments. The first is in the area of gene integration. And the focus is on achieving highest expression level from each inserted vector copy. This can be done by cloning genes into locus control regions and using those for transfer. They express virtually independent of the genome position. Alternatively, a transposase can be used that targets more or less open chromatin. We at ProBioGen have taken this to the extreme by equipping a transposase with epigenetic readers to really find the very best spots in the given host cell. We call this system directed luck. The second area of innovation, I trust, is single cell cloning. One can use sophisticated optofluidic systems or single cell printing to provide thousands of single cells in individual cavities in an automated fashion. Another approach is nanovel-based cloning, where cells, individual nanovels, can talk to each other. This is a particularly gentle approach that allows fast outgrowth of clones. The key question for all those cloning events is whether the imaging that is embedded into the cloning techniques can provide proof of clonality or whether an extra device is necessary for this. There's also interesting new developments in terms of multi-parallel fermentation that is required to analyze clones. 24, 48, or even more fully automated fermenters can be run simultaneously for the best clones while recording critical cell and biochemical parameters. It is amazing how even a deep well today can reproduce the behavior of a clone in a large fermentation system. Another area I would like to mention are intensified processes. They are either applied to the passage before the actual production scale or at production scale. They provide way higher titers based on higher cell densities, but because of the rather stable nutrition of cells, they also provide additional robustness.
And Garima, do you have anything to add to that?
Yeah. So though we know that the timelines are very crucial for a cell line development, but not at any cost. So there has been a balance of trade-off between the cost and timelines, and there has been automation, high throughput screenings, platform medias available. There has been an ever evolving development of expression platforms. One of the examples is our Freedom expicho-s kit, which is having a solution from starting from cloning till the clone selection and also the commercial scale up, because it is tied up with a media which is completely platform and is apt for the host cell line. So I would like to conclude that the field is evolving and we have seen an amazing development here, and timelines have been reduced from a typical 4 to 6 week of single cell cloning to top clone to a typical 5 days by using these optifluidics and other techniques.
I think this is a great place for us to conclude our conversation. I'd like to thank both of our guests for their participation and insight, and I'd also like to thank you, our listeners. If you would like to learn more about stable clone development, please visit thermofisher.com/freedom kit or reach out to a Thermo Fisher representative.